Hi guys, how are you? I miss you. I hope you're taking care of yourself in this critical situation. Today I'm going to explain lesson 2.1, which is about mitosis. I already sent 2 videos and notes. I will send also a worksheet, but I need to explain it and draw it on a paper so you can understand better the phases of mitosis. So basically you have to understand what is mitosis to start. Mitosis is a cell division that happens in animals, plants, and humans. What is the purpose of mitosis? Mitosis is done in order for cells to grow, to repair themselves, to renew. So as I said, it happens in animals, plants, and humans. Of course, we will talk more. We will talk more about mitosis in humans. So I'm going to start with the phases of mitosis. First of all, you have to understand that when the cell is living and is undergoing normal processes like exchanging materials, making new materials, getting rid of waste, getting energy, the cell is living in a phase of interphase. So we say that the cell is in interphase when it's living its normal life. But when the cell needs to divide, it will stop this interphase and it will start with mitosis. Mitosis has 4 phases. Okay, so I'm going to draw it on a paper for you to understand more what is happening in the nucleus of the cell, because the most important thing happening in mitosis is the duplication of chromosomes and then the separation of the 2 cells. Okay, so basically, as I said, I'm going to start with interphase. Pay attention, which is not a phase of mitosis. It happens before mitosis. During interphase, you can see a cell with its cell membrane, with a nucleus, with an organelle called centrosome. It has a very important role that we're going to discuss later on. And inside of the nucleus, you can see chromosomes. But in this state, we don't call them chromosomes. We call them chromatin with an N, because they're not visible. They're not visible even under the microscope. They look like very thin filaments that are uncondensed, we say. Then just before mitosis begins, so when the cell decides to undergo mitosis, of course we're still in interphase. What's going to happen is that the chromosome will be duplicated. Actually, the chromatin will be duplicated. So as you can see, I started with 4 filaments. Also at the end of interphase, I ended up with 4 chromatin that are duplicated. So you can see that we have a copy for each one chromatin, okay? Then the cell will start with mitosis when it's ready, after duplicating the chromosomes or the chromatin. So I'm going to start with prophase, the first phase of mitosis, okay? You can see in prophase that the nuclear membrane, it means the membrane of the nucleus, starts to disappear, okay? And you can see that I have here filaments called spindle fibers. These are the spindle fibers, okay, with 2 asters. Actually, what is this? You remember this centrosome, the organelle centrosome? During prophase, this centrosome will also duplicate and transform into 2 asters. This is one aster, this is another aster, and spindle fibers. These are the fibers, okay? So inside of the nucleus, which is disappearing, of course, you can see the 4 chromosomes. These 4 chromosomes made up of 2 chromatids. We say it's a chromosome of 2 chromatids because it has 2 copies. So one chromatid is 1 copy of a chromosome. And the 2 chromatids, every 2 chromatids of 1 chromosome are joined in something that we call centromere, okay? So now that the chromosomes are ready to be separated, once we're done with prophase, we're going to move on to metaphase. Look at the metaphase. No more nucleus. I have only asters with spindle fibers. And in the middle, I have the lining up of the 4 chromosomes. Okay? Each chromosome is made up of 2 chromatids joined together by a centromere. So the centromere is the one that attaches the chromosomes to the spindle fibers, okay? So we say that the chromosomes are lining up in the middle of the cell to form the equatorial plate. Okay, the equatorial plate. And then we're going to move on to anaphase. During anaphase, the two chromatids of each chromosome are separated, and each set of chromosome made up of one chromatid are migrating to a pole. You see, you have two poles in the cell, let's say north and south. We have chromatids that are migrating toward each pole. You see, so you remember these four chromosomes made up of two chromatids. We need to separate them now. So we're going to separate the two chromatids of each chromosome and send them to different poles. And then we move on to telophase. In telophase, what we're going to see is the nuclear membrane appears again. So we have a clear nucleus. I have the four chromosomes made up of one chromatid in this nucleus, and I have the centrosome. It's back, okay? And I see that the two poles are being separated. You see the two poles of anaphase? Okay, these are the two poles, two new cells that contain a set of chromosome and a centrosome. So I can see that the two cells are being separated. This is what we call the cleavage furrow in the middle, when the cell is pinching inward, okay? So now we're going to separate the two cells. This is what is called telophase. And then at the end, we say that at the end of telophase, we have something called cytokinesis. During cytokinesis, the cell membrane will be totally separated. So now I have two new cells. Each cell has its own cytoplasm, its own nucleus, the set of chromosome, and the centrosome. So the formed cell are totally identical to the mother cell. You see, we started with one cell containing four chromosomes. I ended up with two cells, also each one containing four chromosomes. Of course, these chromosomes will go back to the chromatin stage once we're done with cytokinesis, so we can enter again the interphase. And this is the cell cycle. The cell lives in interphase, then enter mitosis and go through prophase, metaphase, anaphase, and then telophase and cytokinesis, and then goes back to interphase.